Today we're going to look at the light reactions of photosynthesis. And as you can see from this picture, there are in fact two reactions that occur, two parts to photosynthesis. Those are called, get back to my full screen here, those are called the light reactions, which are the ones we're going to look at today. Oh, nice circle, Mr. Monahan. And the other ones are called the Calvin cycle, sometimes called the dark reactions or the light independent reactions. So you can see my little, I thought it was clever anyway, caption up here that says, don't give in to the dark side. That's my Star Wars plug, because we're going to be looking at the light reactions and not the dark reactions. That's my little Star Wars plug. Hope you enjoyed that. I'm going to go away now because I'm embarrassed. No, I'm not. I love Star Wars. Anyway, let's continue talking about this. What we see here, let's get back to photosynthesis, I suppose. What we see here is an overview of the entire process of photosynthesis. And I'll go back to my little screen here so we can see everything on there. As we can see here, there's two reactions, as I said, the light reactions and the Calvin cycle. And what we notice right here in the middle, see how these reactions, okay, so the light reactions are generating products, as you can see here, ATP and NADPH, which are then used as reactants in the Calvin cycle. The Calvin cycle is generating products, NADP plus and ADP, which are then used as reactants in the light reactions. That makes these reactions coupled. Right? One of them produces something that the other one uses, which produces something that the other one uses. They're coupled. They go back and forth with reactants, which is why this process is so cool. Can't have one without the other. Okay, so photosynthesis involves two reactions that are dependent on one another, and today we're going to look at the light reactions. Okay, they're called the light reactions because, surprise, surprise, they involve, oh, look at that circle, that's way better than my first one. So the light reactions because they involve sunlight, or it doesn't have to be sunlight, it can be any kind of light. It can be light from a light bulb, it can be light from your computer, right? Any kind of light, but typically in nature, it's going to be sunlight. So the light is going to hit the chloroplast in this area here, and that's where all the magic is going to happen. There's going to be all kinds of things going on, different reactions that we're going to look at here in a few seconds. They're going to produce some products, ATP, NADPH, and of course down here, oxygen, which is in fact a waste product of photosynthesis. And then those things are going to be used to help power the Calvin cycle. The ATP and the NADPH drive the Calvin cycle, and of course the Calvin cycle takes in CO2, uses the products produced in the light reactions, and creates G3P, which is later used to make organic substances like sugar, but it can be used to make other things like amino acids or fatty acids, anything the plant needs, really. Usually it's sugar, though. Let's move on and have a quick look at what happens inside the leaf. This will be a little bit of review and some new cool stuff. So we talked about, in the last lesson, we talked about the different parts of the leaf. We talked about the cuticle, the epidermis, the stomata. We're not going to go into detail about those things right now. Just remember that most of the photosynthesis that occurs within a leaf takes place in the mesophyll cells. And there's two types. We have the palisade mesophyll cells, these columnar cells here, they look like columns. And we have the spongy mesophyll cells, because they look like sponges. Right. Take a closer look at one of these spongy mesophyll cells, and it will be filled with chloroplasts. If I take a closer look inside a chloroplast, here's what I'm going to see. I'm going to see thylakoids. Thylakoids are the individual units of photosynthesis. Stacks of thylakoids are called grana. As you can see right here, there's a bunch of them in there. If I zoom in using my awesome scanning electron microscope, I can actually see a real picture right here of a granum. Granum singular, granas plural, and right here is an actual thylakoid membrane. Okay, so this is actually, something that I can't take credit for it. Generously donated by these folks here. But basically, yeah, you need a pretty powerful microscope, because remember, we can see, like in our lab, we can see using our microscopes, our light microscopes, we can see the cells. But this is like inside the cell, inside the chloroplast, zoomed right in on the granum. Okay? And this is where the magic happens. This is where the light reactions occur. Light reactions. So the sunlight's going to hit that, and the magic is going to happen. We're going to talk about the, not really magic, but we're going to talk about that now. Okay, so if I zoom in on the thylakoid membrane, then this is not really what it would look like, but this is a picture of what it would look like. We've got the thylakoid. This would be the inside of the thylakoid, and this would be the outside, also known as the stroma. Okay, so the inside, the outside. Now, these thylakoids are engineered. They are built for the absorption of light. They have pigments that trap the light and allow the plant to do certain things. Okay? And we're going to talk about what they do with the light in this case. The pigments that you need to be aware of, there's two different types of chlorophyll, chlorophyll A, chlorophyll B, and they absorb different wavelengths, okay, different wavelengths of light. And we also have the carotenoids. Those are the oranges and the browns and the yellows, the colors you see when the chlorophyll isn't there, like in the fall. Okay? So they have all these pigments. The pigments absorb the light. The light allows these things to do work. We're talking about free energy here. The light would be the free energy, and then allows the plant to do work. Now, what kind of work is it doing? Well, in this case, the plant is going to want to produce NADPH and ATP. And with those, it can eventually produce sugar. Okay? All right. New term, photons. Photon torpedoes, like from Star Trek. It's Star Wars now. I'm into Star Trek. Don't worry, I'm not going to go any further because that would be kind of geeky. But these photons are essentially packets of light energy. That's all you really need to know about them. So they're packets of light energy, and notice these packets of light energy are hitting the chloroplast. They're hitting the thylakoid membrane, and inside the thylakoid membrane there's different photosystems. Photosystem 2, photosystem 1, and then we have an electron transport chain in between those, another one in between the others, and we're going to talk about what happens there. Now, if you remember anything about oxidative phosphorylation, then this process is very similar. There's a lot of similarities between them, and there's some differences too. Okay? So let's move on to the next picture and see if we can't figure out what's going on here. Okay, so remember, photons are packets of light. They're going to hit the photosystems, and that's when the reactions are going to occur. Okay, now notice in this case, we have the chloroplast stroma now up on the top. That would be the outside of the thylakoid, and the thylakoid lumen, which would be the inside of the thylakoid on the bottom. Okay, so this is just a slightly different view, and I just like this picture a little bit better than the other one. I don't know why it's flipped around. Anyway, we'll just start with that. Okay, so let's start with what happens. Let's try and put it in some sort of perspective here. Okay, so light is going to simultaneously, and this is where if you're taking notes, you can start taking notes now. Light is going to simultaneously hit both the photosystem 2 and the photosystem 1. Okay? We're going to look at it in chunks, but it's important to note that light is going to hit those things essentially at the same time. Okay, now photosystem 2, PS2 here, and PS1 over here. Photosystem 2 is also known as P680. Okay, that's another way that you can write photosystem 2, and photosystem 1 is also known as P700. Okay, so those are just different terms. You'll see them. It's important to be able to recognize them. Jot that down so you have those things associated with each other. Okay, so light hits this PS2, which is the photosystem 2, the P680, and that causes some of its electrons, as you can see here, little e, to get excited. And when electrons get excited, they move into a higher energy state. So they have energy. They have extra energy that they didn't have before. So the light excites these electrons, causes it to have extra energy. Now, in oxidative phosphorylation, as part of cellular respiration, we have... So remember in oxidative phosphorylation, the idea is that the excited electrons from the transfer during the glycolysis and the Krebs cycle, the transfer of electrons there, those excited electrons move through an electron transport chain, which helped create a proton gradient. Well, that's essentially exactly the same thing that's going to happen here. These excited electrons created from light hitting the photosystem 2 are going to move through an electron transport chain. And while they're moving through the electron transport chain, their stored energy is going to be converted into kinetic energy. And the kinetic energy is then going to be used to transport protons from the chloroplast stroma to the inside of the thylakoid. Okay, now that's a little bit different. In the oxidative phosphorylation, we're taking the protons out, but in this case, we're moving them in. Okay? As the electrons move through, more protons are brought through, and those protons...
We have drivers, please, drivers.
No, you can't go. It's not time to go yet. Got a few more things to talk about. Okay, so those protons are then eventually going to be used, and we'll talk about where these ones come from in a second, but those protons are going to be used to go through to ATP synthase to help make the ATP. Okay, so again, we're going to store up a bunch of potential energy. Eventually, ATP synthase is going to open up, and those protons are going to rush through. Kinetic energy is going to turn the ATP synthase.
George, to the office, please. Thank you.
And that's going to drive the creation of ATP. Exactly the same as oxidative phosphorylation. But more importantly, let's go back to where the electrons are moving through the electron transport chain. They get to photosystem 1. Photosystem 1's electrons have been excited also by the light, and they move through another electron transport chain where they're picked up by NADP. They reduce NADP to NADPH. Now NADPH, like we said, is going to go over to the Calvin cycle. ATP is going to go over to the Calvin cycle. There's our two products. Let's just go through this quickly one more time again. Okay, so the electrons move from photosystem 2 to photosystem 1 to replace the electrons that photosystem 1 essentially lost to NADP to form NADPH. That leaves photosystem 2 without any electrons. It's got none. Well, I shouldn't say that. It doesn't have the electrons that it lost anyway. So what happens? Well, in an enzyme-controlled reaction, water is split into oxygen and protons. Those protons can help come over here and help produce ATP. But the electrons from the water are going to go through and replace the electrons that photosystem 2 lost. Just to show you a slightly kind of more general picture that doesn't have all the kind of wonky things going on as in the other pictures. You don't see the cell membrane here, you don't see the phospholipid bilayer, you don't see all the different enzymes and things and proteins that are involved. You can just kind of look at it here. So sunlight hits P680, which is photosystem 2. Electrons get excited. They move through the electron transport chain, and as they do, protons move in to the thylakoid. Okay, light also hits the photosystem 1. Electrons get excited. They move through an electron transport chain into NADP, which forms NADPH. The protons that we created here, both from the splitting of water and from the electron transport chain that were brought in, are then used to generate ATP through ATP synthase. That, in a nutshell, folks, is how the light reactions work. Basically, the cell during photosynthesis, the cells that are in the leaf, they want to have some control over the process of photosynthesis, and they need some control. So what they have here are a couple of different mechanisms that they can use to help control the process. Okay? And it's essentially controlled through the concentrations or the amounts of ATP and NADPH. If those concentrations aren't at their optimum levels, then the Calvin cycle can't work properly. Okay? So in order to make sure that these concentrations are within the control of the plant, within the cell, the chloroplast, there's a couple of different pathways here. Cyclic electron flow, okay, is one of those pathways, and then the other one is called linear electron flow. Okay, so cyclic implies that there's a cycle to it, or a circle, and linear, no, just straight line. Okay? All right, so as we said, photosystem 2, electrons get excited when light hits it. Okay, electrons come up here, they move down through. Remember, that's how ATP is generated. The electrons go from a high energy level to a lower energy level in photosystem 1. And as they do that, the proton gradient is created, which goes through ATP synthase to help create the ATP. Now, let's say photosystem 1's electrons get excited, like this, light. Psh, I like the sound effects. They work really well, I find. And essentially, those ones get excited. But let's say we need to generate more ATP. Well, if we don't have enough ATP at that stage, then what happens is that the electrons actually go back through into the original electron transport chain right here. And when they do that, well, then they can be used to create more ATP. Okay? And because this is a cycle, as you can see, it's like a circle. Because of that, it's called cyclic electron flow. Now, let's say there's more than enough ATP that's been produced. Well, then the linear electron flow can occur, and NADP can be reduced to NADPH, which is the normal pathway, the one we talked about before. Okay, so if more ATP needs to be generated, cyclic electron flow occurs. If enough ATP has been created and we need to make some NADPH, linear electron flow happens. So to sum everything up here, folks, the light reactions are used to generate ATP and NADPH, which of course drive the Calvin cycle. Okay? How that happens, first of all, involves electrons being excited by light. Okay? The electrons of photosystem 2 and photosystem 1 get excited by light. The electrons that are excited in photosystem 1, generally speaking, are used to reduce NADP into NADPH. They then go to the Calvin cycle. The electrons excited in photosystem 2 are used through a series of reactions, and electrons moving from high energy level to low energy level to create ATP, okay, through ATP synthase, exactly the same as in oxidative phosphorylation. After the electrons from photosystem 2 have been excited, water is then used to replace those electrons that were lost and create oxygen, which is a waste product, and protons, which of course will eventually be used to generate ATP. And there's a summary. Please make sure you write down the basics of these reactions, and also bring your questions to class. Thank you.